Hi everyone and welcome back to Advanced Higher Biology. Today we are finishing off Unit 2, Organisms and Evolution, and the final key area, which is Part 5, Parasitism. Now, if you have been following these videos live as they've been uploaded, this is actually it. This is finally the end of the entire Advanced Higher course. If you have not been following these live, this is not the end. There are Unit 3 ones which were uploaded just in a bit of a strange order. So again, if you're just starting out, make sure you watch the 3 key area videos of Unit 3 as well. For the rest of you, well done, we're there. Thank you very much for your patience, and let's get on with Key Area 5. So, as always, I'm going to try and get through all of this in one go. It is quite a large key area, so it is divided up. So again, I would suggest you maybe go and do these in little sections, go through your notes, try questions and come back. But let's just jump into it with the first part, 5A, which is a niche. So, first of all, hopefully you remember the term niche going back to National 5 Biology, where we discussed the role that an organism plays in its environment. Now, this, as always, goes into more detail, and there's going to be more classifications of niche with Advanced Higher. So first of all, an ecological niche is what we term a multi-dimensional summary of tolerances. So basically what the organism can take, what it can withstand. So things like the temperature and pH. So what temperature range can they live in? What pH can they live in? And the requirements they require. So the food they need, anything like that, the light that they need of a species. So it's quite a lot of parts that an organism needs essentially to survive in that habitat. Now there's 2 forms here. There is a fundamental niche. Now a fundamental niche is essentially the niche that a species would occupy if there was no other organism available, if there was an absence of interspecific competition. So another National 5 term that's appeared there, interspecific competition, if you remember, competition with other species. So basically, if there's not another species that is also trying to take some of your requirements, then this is your fundamental niche. However, as of always, that's a nice theoretical idea, but in reality, there is almost always going to be interspecific competition. There's going to be another species that is also trying to eat some of the food that you're trying to eat or the water that you want. So in that reality, we have this thing on the right-hand side, which is called a realized niche. So a realized niche is the niche that a species occupies in response to interspecific competition. And the diagrams just kind of highlight this fact here that species B and C verge on the niche. They sort of take over part of that niche as well. Now, as a result of that interspecific competition with other organisms or other species that are also trying to use your requirements, take over your requirements, competitive exclusion can occur. So in this is the idea that if 2 organisms are essentially fighting over a very similar niche, so you eat the same type of food, you hunt the same organism, something like this, essentially one of those species will win. And if that happens, then because you're on the same niche, you have the same requirements essentially, it means that the loser in this case is going to decline. Their population will decline due to local extinction. They are being outcompeted, they are not getting the same requirements, and the winning winning species will actually increase. However, sometimes if those realized niches are sufficiently different, then in that realized niche, potential competitors can exist through something called resource partitioning. So for example, we have a bird here and there's a picture of a tree. In this section here, in the light green, you can see that there's different partitioning of that tree where different species can actually go and exploit those resources. So they're not just all fighting over the one tree, they're essentially able to share. So it's a sort of nice idea of, right, you have slightly different requirements, so we're not all fighting over the one requirement, therefore we can all kind of co-inhabit and live peacefully together. So nice and straightforward with niches, just be aware of the difference between a realized niche and a theoretical niche. Now, we're going to go back onto parasites as this key area is named, which we have discussed beforehand, so just a quick reminder. In parasitism, it's a symbiotic relationship. However, it's a symbiotic relationship where the host is exploited, they lose out in terms of energy and nutrients, and the parasite is going to gain. So if you remember, that's a plus-minus interaction. Now, there's also this idea here of something called reproductive potential. So the reproductive potential of this parasite is going to be greater than the host. This parasite is gaining nutrients, it's gaining energy, it's doing really well, and the host is being exploited. Now, when we talk about these niches, parasites usually have an incredibly narrow or specialized niche. The reason for that is they're very host specific. So a lot of these different parasites, such as the one on the left, the kind of nightmarish one of the tongue-eating parasite that lives in that certain fish, it can't do that to most other species. So it has to actually come across that type of species, that type of fish rather than other species, in order to actually go and eat its tongue and stay there and live and reproduce. So that would be a very narrow niche. They can't just go and invade any other animal. Now, because the host provides essentially everything a parasite needs, they're such a simple and basic function, they are what we call degenerate. So degenerate, along with being a sort of like Shakespearean insult, is a term that means that they lack most of the, say, reproductive structures that you would find in other organisms. They're very simple, they don't really have anything to them at all. So for example, tapeworms don't have a digestive system, they don't have a nervous system or a muscular system. They're missing most of the structures you would find in living organisms. They don't have those because they don't need to digest food. They are just taking the nutrients from the digestive system of another organism. But again, it's this idea that that is a very narrow focus, that very narrow niche that they exploit. But because of that, they also don't need to go and produce or develop any of these systems. They don't require them because they're just mooching off everyone else. Now, there are two types of parasite. This part here is fairly straightforward. You can have ectoparasites, which live on the surface. So we've got a pretty horrifying image of a tick there about to bite and start sucking blood. If you're on the outside, ectoparasite. If you live within the host, though, if you live inside their tissues, like this tapeworm, you'd be an endoparasite. So ecto, out, endo, within. So we're going to go into a little bit of detail about these life cycles now. So we've talked about niches, so we've went a bit more onto parasitism itself, but there's a part here looking at life cycles of two particular parasites. Now, this part is quite tricky. You need to get the order of this correct. We're going to be discussing sexual and asexual reproduction in different structures. These are really common questions in the exam. So I would definitely say this is a part that you should be really focusing on and making sure you know the step-by-step of the life cycles of these parasites. So first of all, again, this was mentioned in higher, but some parasites require only one host to complete their life cycle. However, some of them require more than one host. And there's different words that we use here. You might remember the snail that's been infected by a parasite, essentially trying to trick birds into eating the snail so it can then complete its life cycle in the bird, then continue the life cycle. So if you remember, you have a definitive host, and that's where the parasite reaches sexual maturity. But there also may be an intermediate host, and the intermediate host is the host that is used almost in between. It might require this in order to actually complete its life cycle and get to that definitive host itself. So for example, the snail in the previous image, to then get into the definitive host, which would be the bird. Now, also, we did mention vectors beforehand as well in higher. The idea of a vector is that it's going to transmit a parasite. Now, it might also be a host in its own essence, but a vector is anything that is going to help transmit a parasite from one host to another host. And again, it might also be a host because it might not be aware of this. It might be getting carried in the inside as well. So first off, we're going to talk about Plasmodium. Okay, now this is a parasite which actually causes the disease malaria. You'll probably associate malaria with mosquitoes, but the idea being it's actually the Plasmodium within the mosquito. So if you think that mosquito is a host, but it is also acting as a vector transmitting this, that gives you malaria. So we're going to discuss Plasmodium, how it actually works, how it uses both the mosquito and a human to complete its life cycle. And again, it's worth just knowing the step-by-step parts here. So essentially, you're going to have a mosquito that is infected with the Plasmodium parasite. So this mosquito acting as a vector, it's unaware, it's not meaning to pass on this parasite to you, but it's flying around with the parasite inside it. it bites a human, for example, when it goes to extract blood from you, that Plasmodium parasite goes into the human bloodstream. So we have our first sort of point of contact here. Within the human, there's then going to be asexual reproduction of the Plasmodium occurring in the liver. So liver, first of all, asexual reproduction, okay, that parasite has just went from the mosquito into the human, carries out asexual reproduction in the liver. It will then go and start continuing this in the red blood cells of the infected human. When those red blood cells then burst, we have something called gametocytes are then released into the bloodstream. So at this point, only asexual reproduction has taken place, but we have these gametocytes from the Plasmodium that are now flooded into the bloodstream of the infected human. Now, in this case, if another mosquito then comes across and bites that infected human, the gametocytes then enter the mosquito. So again, this mosquito is sucking up blood. There's now gametocytes within the bloodstream itself. What would then happen is those gametocytes enter the mosquito, and they then mature into both male and female gametes. So the gametocytes giving rise to gametes themselves. Now, because there's male and female gametes, sexual reproduction can now occur. So we now have the sort of fertilization of the male and female gamete. This is taking place in the mosquito. Once this now occurs, the mosquito is then able to go and infect another host, and that Plasmodium is then passed on. So again, it's important for you to know that first of all, there's going to be asexual reproduction in the human, in the liver, then the red blood cells. There's then going to be the burst red blood cells leading to gametocytes flooding the blood vessels of the infected human, and then you need that other mosquito coming across there to go and take this in. There's going to be the male and female gametes in that mosquito, leading to reproduction, sexual reproduction, and the whole cycle can continue. So be aware of the step-by-step parts, where this takes place, which type of reproduction, that sort of thing. The second case study you do need to know is called schistosomes. So schistosomes cause a disease called schistosomiasis, and they reproduce sexually in the human intestine. So this one's different already. It's not the asexual reproduction in the human, it's sexual reproduction. What happens here is once the sexual reproduction has taken place within the intestines of the human, these fertilized eggs are then passed out of the human body through feces, and they will eventually make their way into water. So they'll get into some form of water supply where they develop into larvae, like we can see in this diagram here. The larvae then will go and infect water snails. So if there's water snails anywhere in this water where the so the eggs have passed out of the human feces into the water, they're now in larvae, they infect the snails, and this time asexual reproduction occurs. Now, this produces a type of motile larvae, so a larvae essentially that can swim. So this is going to escape the snail, and it then penetrates the skin of a human. So say, for example, it enters your foot if you're in that water. Once it gets in there, it's going to enter the bloodstream, and again, there's going to be sexual reproduction taking place in the intestines. There's then going to be the larvae stage being released through feces. The larvae then infect the water snails. Asexual reproduction occurs, produces the motile larvae. So again, it's worth just having that sort of cycle drawn out that you know sexual first, asexual, where it takes place, and that's all for schistosomes. Now, we do also talk about viruses in this part here, in this last key area. Viruses are parasites. They can only reproduce inside a host cell. They need to use the host. So they are just, in some ways, an incredible form of a parasite. So, just a quick bit of information on viruses that you do need to know. You've probably came across a lot of this already. But viruses contain genetic material in either the form of DNA or RNA, but they're also packaged in a protective protein coat. Some viruses are surrounded by a phospholipid membrane as well, and that can be derived from host cell materials. So there's a couple of things here. That phospholipid membrane and then the protein coat, that gives viruses a huge amount of protection. And that, for example, is why soap and water is encouraged to break down viruses, because it helps break down the phospholipid coat and things. The other part here, which is really clever though, is that the phospholipid membrane might be derived from host cell materials, so essentially taken from the host. And this is going to be used essentially as a bit of camouflage for that virus as it tries to sneak through your immune system. So first of all, the outer surface of a virus contains antigens, and that host cell might be able or might not be able to detect it as foreign. So normally if something comes into your bloodstream or your body, your immune system is going to basically be able to sense what is on the antigens and either say, this is fine, it's not harmful, or I don't know what this is, let's get rid of it. However, viruses are able to kind of cloak themselves, and sometimes they can sneak past that immune system. So essentially, a quick overview of the life cycle of a virus. So a host cell is going to be infected with the genetic material from that virus. When that happens, the enzymes are going to replicate the viral genome, so the genetic information from the virus. There will then, of course, be transcription of the virus genes, followed by the translation of the proteins of the virus. And then because of that, there'll then be an assembly and release of new viral particles. So it's going to grow and spread very, very quickly. This diagram is actually a really good way of showing this, showing the virus entering. So when we talk about capsid, that's that protective area that we talked about. And there's going to be essentially DNA replication and protein synthesis taking place, and then release of those viral particles to infect more cells. In terms of that life cycle, there's an RNA retrovirus that uses an enzyme called reverse transcriptase. That's going to form DNA, which is then inserted into the genome of the host cell. So that's fuller infection essentially going on. Now, these viral genes can then be expressed in order to then form new virus particles as well. Now, this will be important later on as well, which we will come back to. But for part C, we're going on to transmission and something called virulence. So, first of all, two new words for you. It's very similar to talk about risks and hazards in the first part of Advanced Higher. So basically, if there is a high transmission rate, then there's going to be a greater virulence taking place. So transmission is the spread of a parasite to host. So that hopefully makes sense in terms of the word. So a high transmission rate means there's a very high chance of that virus being spread or that parasite being spread between hosts. Virulence, though, is the harm that is then caused by that parasite. So essentially, if you have too high a virulence, then there might not be a great spread because you could be killing off all the hosts that you have. Transmission, though, if that's high, it's going to spread everywhere, and then that's collectively going to cause more harm. Now, ectoparasites that we talked about earlier on, if you remember, ectoparasites are on the surface of a host. They are generally transmitted through direct contact or by consumption of intermediate hosts. So essentially, something moving from the surface of your skin onto someone else's. Think about a while ago we talked about like head lice is a really good example. Or the consumption of intermediate hosts, they can come across and eat them from your skin, for example. Endoparasites, though, if they're within your body, it's less likely to be transmitted through direct contact. They're often going to then be transmitted by vectors, so a mosquito coming across, biting you, sucking your blood, taking the parasite from within your body somewhere else, or those consumptions by intermediate hosts as well. So if you go and eat something that has the virus inside that tissue, it's then going to be inside your tissues, and you pass it on that way. Now, there are factors that will increase these transmission rates, and the final part of this key area does focus a little bit on just like overall health and well-being and how different situations and living conditions can cause a higher transmission rate. So if there is overcrowding of hosts, so if there's a high density of people, animals, whatever it is, then that's likely going to increase the transmission rates. They're closer together, there's more direct contact, for examples, so a higher chance of transmission taking place. There's also different mechanisms of transmission. So for example, if you have waterborne dispersal stages, then if you have a parasite that's going to spread through water, like we talked about schistosomiasis, if there's people then using all that water and there's, say for example, feces going into the water, that is going to increase transmission rates quite rapidly as well. Now, another thing about parasites, and this is where it gets a bit wild, is parasites are able to modify host behaviours. So often parasites can exploit and modify host behaviours, and bear in mind, parasites usually don't actually want to kill the host. They are using the host. They're degenerate, so they're absolutely reliant on the host on the whole. they're going to use that to maximize transmission. They're going to modify and exploit that behavior to make sure they spread as much as possible. So we have this picture of the snail again on the right hand side. We'll do a quick recap of that. And then on the left, we also have cordyceps. The cordyceps has now become quite popular and quite common knowledge through The Last of Us, this idea of a fungus that's taken over the world. You've maybe seen it in some, say, Attenborough documentaries of this fungus wiping out ant colonies and things. But again, it does modify host behavior, and it does that to maximize its transmission. So some examples of the host behaviors that may be changed include a change in foraging, movement, so maybe it's making them go up a tree to an area where there's a greater chance of being eaten by an intermediate host, or spreading virus particles, fungal particles everywhere. There might be a change in sexual behavior or the habitat that they choose to eat, and also anti-predator behavior. A lot of these are basically putting the hosts in harm's way so that they can be picked up by an intermediate host. Again, the idea isn't just to kill the host for fun, it's using the host behavior or changing the host behavior in order to make sure transmission is at a higher chance. So we have the snail that we talked about before, Leucochloridium, and what that does is, if you remember, it infects the snail's eyes. So it makes them look like this stuff on the right hand side. Absolutely crazy. So those tiny eye stalks that the snails originally had, they're now huge, they're now green and white and sort of pulse in a really strange way. They look like sort of massive caterpillars. So these caterpillars that were just going around, these snails that were going around minding their own business, it looks like they have two massive caterpillars now on their head that will often go and put them into an area that they're more likely to be seen by birds. The birds come across, eat the snail. By eating the snail, the Leucochloridium parasite is then within the bird. The life cycle takes place, and then the bird poos out some of the eggs. Those eggs are released in the feces, and snails come across, eat the bird poo, and then the whole cycle just continues. So the whole idea is that it's putting the host in a vulnerable spot in order to go and complete its life cycle. You don't need to memorize this example, it's just quite a good one, it's quite a common one that comes up as well, but this isn't one of the case studies that you do actually need to know. Another word that does come up here, another term rather, is extended phenotype. So again, going back to National 5 again, phenotype, the physical characteristics of a species. An extended phenotype is essentially if a parasite changes your behaviour, and often it'll actually sort of change part of you. Say for example in that snail, the eye stalks looking like caterpillars, or in cordyceps you've got bits of fungus literally erupting out of you. That's now essentially changed what you look like or what you are. So that's what we call the extended phenotype of the parasite. So the host behaviour is now essentially a part of that parasite. So something just to be aware of. Again, just some further examples of some host behaviours that can be changed. And again, they are changed in order to try and maximise that transmission rate. Is there's a toxoplasma parasite. And this was a study that was done years ago that showed if you are a rat and you're not infected by toxoplasma, if you smell cat urine, then you're pretty scared of that because you're likely going to be killed and eaten by a cat. That's a very natural thing in the animal kingdom to be afraid of. However, as soon as a toxoplasma infection takes place in a rat, they actually seek out cat urine and they will sort of put themselves in harm's way after smelling cat's urine. The idea of that is that a cat is actually the definitive host of the toxoplasma parasite. So the toxoplasma wants the rat to be eaten by the cat so that it can move into the cat system, complete its life cycle, and then its eggs can be pooed out. Again, really fascinating that it's been able to change the host behaviour of the rat. The rat is now infected by the toxoplasma, basically realises there's a cat nearby and thinks, oh brilliant, please eat me. So again, a huge change in the host behaviour. It is going to kill the rat, but it's killing it with the purpose of completing the life cycle, passing on its transmission into the cat. Some things that also might be changed as well is things like the immune system of a host. Usually it's quite weakened. It might be the size of the host, reproductive rate, overall lifespan as well. But again, like I keep saying, the whole point is these modifications are there in order to benefit either the growth of the parasite, its reproduction, or its transmission. It is there to benefit the parasite. It's not just killing the host. It needs the host, it's using it, and it's changing its physiology in order to benefit the parasite itself. So if we then go on to defense against parasitic attack, okay, we don't want parasites. They are very, very good at infecting us. And if they have any of these methods of trying to evade your immune system, or a clever way of getting into an animal or something like this, it kind of goes back to the Red Queen that we talked about at the start of this topic as well. Parasites are evolving all these weird and wonderful ways to infect us or infect animals. Animals are finding a lot of amazing ways to try and avoid them or get rid of these parasites. So first of all, going back to some fairly basic ideas in biology, mammals have a bunch of immune responses to try and protect us from any form of pathogens, and they can be non-specific and specific. So some examples of non-specific defenses are physical barriers like your skin or the secretions from your eye or your mouth and things like this. We also have inflammatory responses to infection, we have phagocytes, and we have natural killer cells. Now, these are all just general defenses, therefore non-specific. They're not there for any one reason or against one specific type of pathogen, parasite, disease, virus, whatever it might be. They are just there to try and essentially protect us from most things. Now, for those examples, just go into them a little bit more detail. So your skin, your epithelial tissue, that's there to basically just block parasites just walking straight into your body, into your internal environment. There's hydrolytic enzymes in your mucus that help break down things. Your saliva and your tears are able to destroy the cell walls of bacteria. Areas like your stomach, vagina, sweat glands, they all have secretions to allow low pH environments. The idea of those low pH environments is to denature proteins of pathogens. So killing off those pathogens, preventing them from establishing an area and spreading. It's all there in a non-specific way to try and make any areas of your body essentially uninhabitable area for pathogens to take place. Other things as well, we have signaling molecules. So if you have an injured cell, they will release molecules, and that leads to enhanced blood flow to the site. If there's more blood flow going to an area, there's going to be more antimicrobial proteins, and there's going to be phagocytes in there as well. If you want a little reminder of phagocytes, there's quite a lot of Nat5 that's appeared in this key area actually. They are used to kill parasites through phagocytosis, apologies, and they use enzymes contained in lysosomes. So if you remember, the parasites are engulfed in a vacuole, they're broken down and destroyed through this process of phagocytosis or phagocytosis. So again, if you have a lot of phagocytes being brought to an area through that increased blood flow, it's increasing the chances of anything that shouldn't be there being engulfed and destroyed through that process. You also have natural killer cells as well, and they're able to identify and attach to cells infected with viruses. We've talked before about them being able to kind of notice what's foreign and what's not foreign. They're going to release chemicals that lead to cell death, essentially by inducing apoptosis within that cell. So they come across, they bind to a cell that they know shouldn't be there, and they essentially get it to self-destruct. Nice and easy, but again, still non-specific. They're just going around doing that to anything that should not be there. In terms of specific cellular defences though, you also have a range of white blood cells that are constantly going through your bloodstream, kind of looking for anything that's kicking off. If any tissues become damaged or they become invaded by something, these cells release cytokines. These cytokines, again, increase that blood flow to the area that will bring across, as you mentioned, antimicrobial proteins and phagocytes, so non-specific responses, but it will also bring specific responses. So specific white blood cells will come across and accumulate at that site. Now, in terms of lymphocytes, mammals contain a lot of different lymphocytes, if you remember, a type of white blood cell, and they all have different receptors on their surface which can potentially recognize a parasite antigen. So these lymphocytes come across, they have their receptors, they're going across to something that's been identified, and they're trying to figure out, is this a parasite antigen, or is this something that's okay? So they come across, they find out what's going on. The binding of an antigen to a lymphocyte receptor selects that lymphocyte to divide and produce a clonal population of that lymphocyte. So in this this diagram on the right hand side, it's a good way of showing that antigen X has bound to this X lymphocyte. A, Z, and B, and Y, for example, does not bind. It's not specific, so they are not required. But the X antigen or the X lymphocyte is now just going to reproduce, so you have more of those lymphocytes, as they are the ones that are needed because they are specific to that antigen. Then what happens is some of these selected lymphocytes will produce antibodies. Others can induce apoptosis, like we talked about earlier on, in these parasite-infected cells, or they'll produce antibodies to destroy them as well. So as we came across before, they're going to come across, find out what this is, and they're going to increase in number and destroy these cells. Excuse me a sec. So antibodies possess regions where amino acid sequence varies greatly between different antibodies. That variable region is what gives the antibody its specificity for that binding antigen. So that's the one little difference that makes all of these different antibodies unique and are able to actually be specific for that antigen. When that antigen binds to that binding site, the antigen-antibody complex forms, can result in the inactivation of the parasite. It basically can render it useless, stop it from doing anything. Now, that can then mean that a phagocyte comes across to go and undergo phagocytosis, and or it could stimulate a response that destroys the cell, going to cell lysis as well. So again, the whole point being, find this, render it useless, kill it off. Now, one thing as well, and you'll have came across this before in biology, I'm sure it's actually quite commonly a problem-solving question, is this idea of a primary immune response when you're initially exposed to an antigen and a secondary response. So we mentioned there about how these lymphocytes come across, basically see which one fits the antigen that they've came across. Obviously, for that to happen, they'll have had to have came across that antigen before in the first place, for them then to go and reproduce and use the correct lymphocyte for whatever they've been exposed to. So that initial exposure to an antigen produces memory lymphocytes. So memory lymphocytes are specific to that antigen, and they produce a secondary response if the antigen enters the body in the future. Now, if this occurs, the speed of antibody production, concentration, and duration are then increased. So you can see here, you first of all have an antibody concentration that's fairly low. They've came across that antigen, it builds them up. Then it'll decrease over time once the infection is gone, but there will be some memory lymphocytes kicking around in case it appears again. As soon as it sees the antigen for a second time, the response, like we said, is much quicker. There's more antibodies, there's a bigger concentration of antibodies, and it's there longer. And then on to the final part of this key area. So this is it, the last slog. We look at evading the immune system. So parasites have evolved ways of evading the immune system, as you have said. Endoparasites, so within you, are able to mimic host antigens and able to evade detection. So all that we've just talked about there with detecting antigens, if you find a way of tricking that system, it means you can bypass those specific defenses and establish yourself in the spread. Now, this is a way of modifying immune responses, reduce your chances of destruction. So, antigenic variation in some parasites allows them to change between different antigens. So if they go into a host, they can change their antigen, and it can also allow reinfection of the same host with a new variant. So although you're the same pathogen you've came across before, it's essentially changed its coat, it looks different, so you don't recognize them. Now, some viruses can escape immune surveillance by integrating their genome into host genomes, then existing in an inactive state. This inactive state is known as latency. So basically, it comes into the immune system, it integrates its genome, and then it just sort of stays quiet for a while. And then when favourable conditions arise, it becomes active. Now, this actually is the last part. I forget this goes on to F, but one of the main parts we come across in terms of diseases is the challenges in actually controlling and treating these. So we look at epidemiology. Epidemiology is the study of outbreak and the spread of infectious diseases, and epidemiologists are the people who work within this. So first of all, one thing you may came across, especially during COVID, is the idea of herd immunity. So the idea of herd immunity is that not everybody in a population needs to be either immune or vaccinated against something for an epidemic to be prevented, but you do need a threshold, you do need a certain number. So for example, down here in the bottom left, if there's no immunity, then whatever disease is happening can spread from everyone. However, if you have a significant population or significant portion of the population that are immune, then that prevents it being passed across as much. There might be some people that could be susceptible to this, but the chances of them being in contact are so small. So the herd immunity threshold is the density of resistant hosts in the population that are required to prevent that epidemic, that breakout. Now, in terms of designing vaccines, we talked about antigens there and what some of the issues are. Now, vaccines themselves contain antigens, and those antigens are going to cause an immune response. This can cause issues because if there's a disease that has antigenic variation, that has to be reflected in the design of the vaccines. So if it's not always just the same antigen, it's very difficult to then just give someone a vaccine with the antigen, because if the other variant comes across, there's no response to it. Some parasites are also very difficult to grow and culture in a lab, making it difficult to design vaccines. A really good example of this is the common cold. Everyone's always talking about why do we not have a vaccine for the common cold. As you may know, it essentially changes its antigens every year. So if you get a flu vaccine, the flu vaccine you had in 2025 will be different from the one in 2024, and that's to reflect the difference in the antigens that year, and there'll be a different one next year. Now, the similarities between host and parasite metabolism can also make it really difficult to find a drug that's only going to target the parasite, because they are so heavily combined and they overlap so much. You don't want to have a medicine or a drug that's going to target part of, say, the human body as well, but also kills the parasite. You want to only affect the parasite, so it's difficult to make these up. The other challenges as well in terms of trying to actually control and treat any form of epidemic or spread of disease is that parasites are able to spread rapidly, especially in the areas of overcrowding, and tropical climates are just perfect in terms of temperature and humidity, water. They're very susceptible to diseases. These overcrowdings can occur in places such as refugee camps, so any form of war, natural disaster. The next knock-on impact of that is going to be an overcrowded area that's going to result in a spread of diseases. It's going to cause a lot of problems, especially in what we call less economically developed countries as well. All of these conditions make coordinated treatment and control programs difficult to achieve. You're going into a bad situation with a lack of sometimes sanitation, you have overcrowding, diseases can be rife, and it's very hard to actually just go and treat this. So sometimes the very basic levels involve clean water, hand washing, this sort of things. So a lot of projects involve sanitation combined with vector control, and sometimes that's the only practical way of actually controlling these. Now, we talk about this in modern times, but this has always been the issue. So a good example of this is the plague in Edinburgh. When you look at essentially where everyone lived along the Royal Mile, there's just all these people living cramped and tiny, dark houses, poor sanitation, houses above and below you. Any form of disease of any form is just going to spread so quickly through those areas, particularly if it's not sanitary as well. And finally, one of the big improvements in parasite controls reduces child mortality, and they result in population-wide improvements in child development and also intelligence, as individuals then have more resources in order for growth and development. So the whole idea is this benefits everyone, and as I said, sometimes the most practical solution is the basic ones. It's not just firing in vaccines, it's making sure everyone is drinking safe water, people aren't bathing in areas that are contaminated, all these sort of things as well. And that is it. That is the end of advanced higher biology. So as I said before, if you're watching this maybe in 2026, 2027, please don't think that's the end unless you have also seen Unit 3. There is a shorter unit as well. But if you've been watching this live as it's been uploaded, that is the last video. And as you can tell, quite luckily, because my voice is about to go. So if you have made it through all of this, just a very quick thank you to you. I know a lot of you have been asking for these, and it's taken quite a long while to make them. But I do really appreciate you sticking with these. I do really appreciate all your comments, and particularly for people who have been watching these through all of National 5, Higher, Advanced Higher. It's crazy thinking when I first made this up for my classes that there's now 10,000 subscribers and you're all using this. So it's been amazing, and I really hope that you have found some uses through these. And as I said, particularly if you have used them for the last 3 years, that's a huge amount of your education. I really do wish you the best with your exams and whatever you go on to do next. So again, please spread the word, pass them on to people who might find them useful, and thank you so much for all your comments. And that's bye for now, and I won't see you again for the next video. So very good luck for everyone, and hope that exam coming around the corner works out well.